for your nucleic acids. Now, to formally start our discussion, we formally define nucleic acids as polymers that we call polynucleotide. And of course, if si carbohydrate ang monomer niya ay monosaccharide, si proteins amino acid, for nucleic acids, our monomer is your nucleotide. Alright, so ayan class, ang overview of your nucleic acids. Source natin, main source is meat, seafood, and legumes. Class, I will emphasize the feature. Similar with carbs and proteins, they are polymeric and chiral. And as I have discussed, diba, we also have classifications based on the structure. For nucleic acids, we have two. The deoxyribonucleotides is the V monomer of DNA, and ribonucleotides for RNA. Alright, and then of course, bond type, again, phosphodiester bond. In organic chemistry, ang nature ng bond na to is ester. Alright, from the name itself, diester bond. And lastly, very unique po, si nucleic acid in terms of its key function. Because comparing it with the first three biomolecules, they have energy function. For nucleic acids, ayan, they are used for genetic information storage and genetic transfer. Alright, so I hope that's clear guys. Again, si nucleic acids, it only participates in information storage and transfer. So now, we will proceed with the composition of your nucleotides. Alright, so first one of course, this is the structure of one nucleotide. Ayan, so we have a nitrogenous base, a sugar. So your sugar depends whether your nucleotide is deoxy or just ribonucleotides. Okay, and lastly, all nucleotides have phosphate groups. Okay, so yung nitrogenous bases, they can be classified into two categories. We have purines and we have pyrimidines. For purines, we have adenine and guanine, and for your pyrimidines, we have cytosine, uracil, and thymine. Okay, so later on, we will see that these pyrimidines are distributed on RNA or DNA. Okay, not all of these pyrimidines can be found in DNA. So class, in order to help you in familiarizing this nitrogenous base, ayan, we have a mnemonic. For purines, pure Ag, diba? Pure silver, kalaban ni pure gold. Alright, so for pyrimidines, we have prime cup. Okay, class, please remember this mnemonic. It's very important. So sa board exam, may mga questions, typical board exam question, which of the following is not a pyrimidine? Okay, sayang ang 1 point if you do not know the correct answer. Alright, so class, again, going back, this is the structure of your nucleotide. But first, we also have a term, nucleoside. So that's the component of nucleoside, sugar and base. Okay, wala po siyang phosphate group. When we added the phosphate group, we now call them nucleotide. Ayan, so of course, later on, you will see that your pyrimidines and purines participate in base pairing in your DNA. Okay, but now I would like to highlight or emphasize that the backbone of your nucleic acids consists of your sugar and phosphate only. Okay, hindi po kasama ang nitrogenous base pairs. Again, ang backbone is the alternating sugar and phosphate backbone. All right, so this is again the reiteration of your nucleotide structure. We have a sugar, a phosphate, and nitrogenous base. Ayan, so class, check your manual. We listed the structures of your ribonucleotides and deoxyribonucleotides. So let's briefly discuss the difference in their naming and symbols. Okay. For ribonucleotides, when it is a nucleotide, we end the name in suffix ate. For instance, for adenine, na nitrogenous base, we call it adenylate. For guanine, guanylate, uracil, uridylate, and then of course cytosine, cytidylate. Or of course, it also has a long name. Alright, for symbols, for your adenylate, A or AMP, for guanylate, G or GMP, and of course, the same structure or pattern for your symbols apply to your uracil and cytosine. Okay, now, sir, paano pag nucleoside lang? Of course, we will end it in INE. Okay, for adenine, adenosine, for guanine, guanosine, for your uracil, uridine, and for your cytosine, cytidine. I hope that's clear. For deoxyribonucleotides, we will just add a prefix, deoxy, because if you will examine the structure at the C2 position of your sugar, class, remember our discussion on carbohydrate, this is a ribose sugar. Ayan, so as you can see here, in your deoxyribonucleotides, ang sugar niya is deoxygenated at C2 position. Okay, and this is really important because it makes your deoxyribonucleotides relatively unreactive compared to your ribonucleotides, contributing to the stability of DNA compared to your RNA. Okay, so I hope that's clear. Again, ribonucleotides and deoxyribonucleotides are both monomers for forming polynucleotides. Okay, so now we will now compare your DNA and RNA. So class, again, sa DNA, it is made up of deoxyribonucleotides, while RNA is made up of ribonucleotides. In the location, or for their location in the cell, sa DNA, it can either be nuclear or mitochondrial. Yes po, your mitochondria has its own DNA, has its own genetic information. Because later on, you will see maraming ganap sa mitochondria. Masyadong maraming ganap sa mitochondria. Okay? So for RNA, it can either be nuclear. So because yung DNA replication, it occurs in your nucleus. That's why, kapag nagkaroon ng transcription, the RNA produced is nuclear in terms of its location. Okay? So guys, again, that's just a glimpse of our discussion on central dogma. Okay? It can also either be cytoplasmic because ang translation, it occurs in your cytoplasm. And we know RNAs take a crucial part in our translation process. Alright? For DNA, function, it's very static, meaning for information storage lang siya. And as I have said, si RNA, dynamic siya because it has several roles in translation, transcription, as well as or even in regulation. Alright? For its structure, si DNA, as we all know, double-stranded po siya, contributing to the stability of its structure. However, si RNA is only single-stranded or minsan, it is in complex shape. So later on, I will flash some examples of RNAs. Okay? For structure, again, it is in anti-parallel helix, while your RNA, it can form different secondary structures such as your hairpin and your loops. Alright, so class, I will just reiterate, ang sugar po ni DNA is deoxyribose, meaning deoxygenated at C2 position. And lastly, for nitrogenous base, we have AGCT for DNA. Si thymine kay DNA lang, while si uracil kay RNA lang. Alright, and of course, again, ang stability, much more stable si DNA compared to RNA because of its structure and strand formation. O, diba? To compare them, ayan, if you are watching or if you already watched Game of Thrones, so ayan si Daenerys, so you can see in her hair, ayan si RNA, single-stranded, si DNA, double-stranded. O sir, patingin naman kami ng example ng secondary structures ng RNA. Okay class, this is just a supplementary discussion. As you can see, very dynamic po ang structure ng RNA, and thus, of course, yung functions niya din ay dynamic. It is varied. As you can see, it can form double strand, a bulge, hairpin, asymmetric internal loop, or coaxial stack. Okay? Very diverse depending on the localization of your RNA in the cell and its specific functions. No need to memorize the secondary structures. Clear ba, class? Ayan. So now, we will proceed with the discussion of the unique bonds in your nucleic acids. That is your phosphodiester bonds. So again, ang nucleotides natin, they are joined by phosphodiester linkages. And similar with your carbohydrates and your proteins, it also has directionality. Okay? So between the 5' phosphate group of one nucleotide and the 3' hydroxyl group of the next. Okay, so this is the phosphodiester linkage. And very clear po ang directionality ng nucleic acids natin. That's it. That's from 5' end to 3' end. Okay, so this one, class, this just shows the complementary base pair. So this is evident in your DNA, your deoxyribonucleic acids. Again, class, for your directionality, we have 5' because of the free phosphate group and then 3' because of the free hydroxyl group. Okay. So now, we will focus more on the properties and the processes or concepts that are related to your DNA, your deoxyribonucleic acids, because relatively much more complex po kasi ang structure ni DNA compared to your RNA, although static lang ang function niya. Okay? So here are related concepts for your DNA. Alright? So as we have discussed, its strand and its structure is antiparallel helix and it's double-stranded. And that base pairing is based on Chargaff's rules. O, diba? I am expecting that you are familiar with this rule. Meaning lang class ng rule na to, is that the concentration of A is equal to the concentration of T. And then the concentration of G is just equal to the concentration of C. So yan po ang specific base pairing natin. A is to T, G is to C. Dapat laging consistent that A and T have equal concentration in your DNA as well as G and C. So for your Chargaff's rules, we have A plus G must equal to T plus C. Okay? So medyo confusing siya, class. So ang tip ko dyan is just memorize the Chargaff's rules base pairing. Okay? A is to T, G is to C. For our complementary base pairs, the stabilizing interaction is your hydrogen bonding present in your nitrogenous base. All right. So later on, I will emphasize where can we found that specific hydrogen bonding interaction. Okay. Aside from base pairing, class, we also have other stabilizing interaction. So in DNA, we have an interaction we call base stacking interaction. So yung interaction naman na to is perpendicular to your helix axis. Okay. It's the interaction of your base above and your base below the helix. And of course, this is also enhanced through hydrophobic shielding because as we all know, ang nitrogenous bases they have regions of hydrophobicity. Okay. So lastly, I think this is the last concept. So we also have grooves in DNA helix. So we have two types of grooves, major and minor grooves. Okay, and this act as binding sites for our different central dogma processes. Okay, I'm sorry, class, meron pa pala tayong final last. Ayan. For directionality again, okay, so all phosphodiester bonds have the same orientation. Okay, don't be confused ha. Ang directionality ng ating DNA is 5' to 3' po. Okay, but enzymes read the transcript from 3' to 5' position. Okay, so class, again, this is the structure of your DNA. O sir, nasan po dyan yung base stacking interaction? So this is between this base pair and this base pair. Okay, so again, kasi perpendicular sila sa helix axis. Alright, so again, that's your base stacking interaction. For your major and minor groups, we have here the major groups are relatively large surface area. This acts as a major binding site in our central dogma. Okay, so syempre need natin magbind ng enzymes in order to start our central dogma. And of course, minor groups can also serve as binding sites. Alright, so our next topic is on the history of DNA discovery, which is actually quite controversial. Okay, so let's just read the statement. In 1953, James Watson and Crick proposed the double helix model of DNA. Their discovery was based on Chargaff's rules of base pairing and X-ray diffraction images of Rosalind Franklin and Maurice Wilkins. And of course, because of the data of Franklin and Wilkins, they were able to conclude that DNA really consists of two strands twisted into a right-handed double helix. So very controversial because even PRC, they acknowledge the answer Watson and Crick as the one who proposed the double helix model of DNA. So I think this already appeared in the board exam. Okay, again, let's acknowledge that the data, the diffraction images of Rosalind Franklin and Maurice Wilkins has a big part in unfolding the structure of DNA. Okay, but for the sake of our board exam preparation, again, ang ino-honor po is si Watson and si Crick. Okay, so I hope that's clear, class, ha? So now, let's proceed with our last subtopic. Ayan, sir, malapit na tayong matapos. Yes. So now let's first discuss the numerous function of ribonucleic acids. Diba, as I have said earlier, very dynamic si ribonucleic acids. But no need to memorize their specific functions. Okay? So hindi naman siya ganun irrelevant. But here I would like to emphasize that you need to be familiar with these three main ribonucleic acids. mRNA or your messenger RNA, tRNA, your transfer RNA, and ribosomal RNA or your rRNA. Okay? So we will discuss this kasi more frequently sa central dogma. Okay, so aside from this three, we also have your small nuclear RNA for splicing. Okay, it has a crucial role in splicing. We also have your SNO RNA or your small nucleolar RNA. All right, we also have a microRNA for gene expression, small interfering RNA still for gene expression, and long non-coding RNA for gene expression as well. Okay, so again, class, I would like to emphasize this tree should be memorized. Okay, at least you are familiar with mRNA, tRNA, and rRNA. Okay, so let's now proceed with the other functions of your nucleic acids, particularly your NTPs. Sir, ano ba yung NTPs? NTPs are your nucleoside triphosphates. And upon hydrolysis of this molecule, it can provide chemical energy. O diba, sikat na sikat si ATP or your adenosine 5' triphosphate because this is the central carrier of our chemical energy in biological system. But upon discussing our metabolic pathways, we will see as well that GTP, UTP, and CTP are also used, okay, for some reactions. All right. So this is the structure or the general structure of your nucleoside triphosphates. From the name itself, it has three phosphate groups. So yan ba yung structure ni ATP? Yes po. Ang base niya here is adenine. All right, so now let's proceed more on discussing several types of your coenzymes that have nucleic acids in their structure. So for instance, we have coenzyme A. We have your NAD plus or your nicotinamide adenine dinucleotide, and we also have your FAD, your flavin adenine dinucleotide. All of them have adenine in its structure, or adenosine rather. Okay, so of course this is very important. The structure of this tree, as you can see, have an adenosine in their structure. Class, in our discussion in carbs metabolism and lipid metabolism, we will encounter these three coenzymes more frequently. Okay, so in addition, ayan, some nucleotides regulate cell signaling, such as your COMP, your cyclic AMP. Okay, so this is produced from ATP by adenylyl cyclase, and this is a common second messenger. Okay, so again, class, the cyclic AMP participates in signal transduction inside our body. Okay, so class, konti na lang, we have another example of your nucleic acid, si coenzyme A, and of course, ang famous na structure ni acetyl-CoA, diba? So this will be discussed more sa ating metabolic pathways. Bida siya sa metabolic pathways, okay? But for now, let's briefly discuss or introduce your acetyl-CoA in terms of its structure, alright? So acetyl-CoA, again, it participates in all metabolic pathways of your carbs, lipids, and amino acid. O sir, ano po ang main purpose ni acetyl-CoA? Of course, taga-deliver, o diba? Parang JNT siya, delivering the acetyl group or the acyl group. Ito siya, class, colored or boxed in color blue to your citric acid cycle. Okay? So of course, this will be oxidized for energy production. Alright? So ang backbone po ni acetyl-CoA is this thioester functional group boxed in red. Alright, so again, this is the structure of your coenzyme A from our previous slides. Ayan, so now class, we have a did you know segment. Ayan, did you know that your DNA is longer than you think? So of course, class, because of the structure of our DNA, it seems short lang inside our system. But every single one of our cells holds over 3 billion DNA letters. Napakarami yan. And if it's stretched out, that's nearly 2 meters long. Okay, so our body contains about 37 trillion cells. So the total DNA could stretch to the sun and back over 300 times. So of course, very efficiently ang packing ng DNA natin inside the nucleus. Okay, so the tightly packaging is very important to maintain the stability of our DNA. Again, because it's our genetic information storage. Alright, so last, did you know? Humans share about 60% of their DNA with bananas. Diba? Even though that we're vastly different from bananas, all life shares a common genetic language. Okay? So that's why, parang more often than not, we are being compared with bananas or other organisms such as cats, your strawberries as well as chimpanzees. Diba? We are mostly related or similar with chimps or chimpanzees. Okay, so class, that's it for our discussion of nucleic acids. And with that, I will have my final Lars words to conclude our discussion. So this is the summary of your nucleic acid. Monomer, nucleotides, check. Directionality, again class, I will emphasize, this is the directionality 5' because of the free phosphate group at the 5' position, and then the 3' position has a free hydroxyl group. Okay, naturally occurring forms, as we have said, right-handed po siya, or we call it your BDNA. Okay, class, remember BDNA because we also have ADNA and ZDNA, but we will not discuss it here in our CC. Energy function, again, none because it's for genetic information storage. Second to the last, digestion mechanism. So we have nucleases as the main enzyme, and the site of digestion, of course, similar lang sa lipids, and of course proteins, small intestine. And lastly, solubility, we have water soluble because of the presence of numerous hydroxyl groups. First, in the sugar component, and second, the charged phosphate group. All right. For structure and composition, ayan. So we have nitrogenous bases, purines and pyrimidines, and we also have your nucleosides and nucleotides. Again, class, ang difference lang between the two, si nucleosides ay walang phosphate. Okay? So next, we have your classification, DNA and RNA. Recall, DNA, static function, RNA, dynamic function. DNA, more stable, RNA, less stable because of its single-stranded structure. Okay? And notable examples that we discussed for RNA, the 3 RNA molecules that participate in your central dogma, and of course, the adenosines. Okay? Those molecules that have adenosines in their structure, like your ATP, CoA, NAD+, FAD, acetyl-CoA, and cyclic AMP. Alright, so to summarize our discussion on biomolecules. So first, here is the objectives that you need to brush up. Okay, so again, natakal natin lahat to. This is our review checklist. First one, we identified correct monomers and bond types. We also recognize the different structural features and directionality of all of your biomolecules. And of course, again, class, ang bida sa discussion natin is their structure and function. Okay, and we also link each biomolecule to their digestion mechanism, particularly the enzyme responsible and the site of digestion. Alright, and then of course, yung solubility, very important yan, as well as their metabolic energy roles. Okay, class, as I have promised earlier, here is the summary of mnemonics. Okay, please find some time to memorize the mnemonics. Very important yan for you to efficiently memorize or familiarize some important concepts in biomolecules. Okay, this involves pentoses, hexoses, your saturated fatty acids, your vitamin B complexes, ayan, essential and non-essential amino acids, your protein and your enzyme classifications, your inhibition, particularly reversible inhibition, and your enzyme activity. Okay, so that's it for our crash course number 4. Alright, class, please answer PT number 4. So this is all about nucleic acids. Okay, this is 10 items for 15 minutes. Closed manuals ulit tayo, and of course, I will discuss the answer in a separate video. Okay, see you on our next crash course. Bye!